Okay, so in this video, we're going to focus on the method section. Other terms often used for the method section can be materials and methods, methods and materials. I'm wondering what else I've seen, but basically describing how did you conduct your study, ultimately then, giving the background of how you came to your results. So that's the purpose of the method section. You want to provide a clear overview of what you and your research team did to get to your results. And I say research team here because rarely will you publish by yourselves. You also want to make sure the method section gives other scientists the information to ultimately replicate your study. So this is a little bit of a balance act because you want to include enough information to do so, but you also don't want to give unnecessary details, right? And so sometimes that's hard to do. I see a lot of feedback from editors. They either say, oh, there's some information missing, needs to be more detail, or this is far too much. So it's a little bit of a balance act, and this is why it's helpful to get other people on your team and colleagues review your work. If they think this is adequate, then you're probably in good shape. You also want to make sure you organize the information for your reader, and the number one thing to do that is to actually divide your method section into subheadings. And I'm going to give you some examples of subheadings. So you can have a paragraph or two per subsection in the method section. It's really helpful because just looking over the headings sometimes can give a reader an idea of what your study design looks like and how you arrived at your results. You want to also make sure you cite commonly used methods instead of writing about them in great detail. So if there's a research article that, or another article that describes this particular method in great detail and you're using the same method, just cite that. And if you make any changes, you got to make sure you note that. But in general, if you're citing a widely commonly used method, no need to explain it in great detail. That can save you time and space for other aspects of your methods that you want to provide some detail for. And use a flow diagram or table when possible. Now, I've seen this mostly in studies, usually in articles based on clinical trials with a large population. So I'm actually going to show you that flow diagram of that study from drug safety that we were looking at in our introduction video, or a video in the introduction section, which makes sense because there were over 700,000 people in that study. And lastly, this is actually a section where you can use passive voice and you can use jargon, very discipline-specific terminology, more so than in other sections, because the method section is really the section where you got to show that you know your stuff in your field, and you can show off with jargon. And passive voice, we'll talk more about active and passive voice next week or the week after, I think in the last week, when we look at sentence structure and style. But in general, passive voice, try to avoid it in the narrative sections, in the intro and discussion. But here again, it's okay because there's only so many ways you can say in active voice that something was done. The survey was administered. You don't want to say, we administered the survey, we did this, we did that. I'm going to show you some examples though where you can actually be really creative with active voice as well. So, let's look at the next slide. Common areas to cover in your method sections, the materials, the things that you've used. If you have participant subjects, who were they? How did you recruit them? What's your experimental protocol? What's your study design? What kind of measurements, dependent, independent variables, did you rely on? And you also want to provide some analysis, but not interpretation of the data. You want to leave it to the results section. Very important. Do not interpret what the data means that comes in the results. And ultimately, and then the larger context of that comes in the discussion section. So often what I see from editors and from reviewers when I work with writers and I see the reviewers' comments, it's often like, gee, this should have been in the results section. It's not necessarily the end-all be-all for your article, but it's good to train yourself to really differentiate that. I've included this table here. It's from an article by T.M. Ainsley on who, what, when, where, how, and why, the ingredients in the recipe for a successful method section. And he basically talks about the who, the what, the when, the where, the how, and the why, right? And those are all questions you can ask yourself as you're writing your method section. You will have access to these PowerPoints on the Moodle platform as well, not just the videos, but also the actual PowerPoints. So you can always go back to this slide. And if you're interested, I'm going to actually post a link to this article in the Moodle section as well. It's a really interesting article. Here's a couple of other articles on the other sections of an IMRED article that I think I have as well. And if I find them, I'll post them there as well. So, you can read that in your own time. Again, great overview. I'm not going to go into great detail here. You can review that later on. But again, just keep in mind who, what, when, where, how, and why. And why meaning not so much what's the meaning of the of why what you're finding, but it's the why are you choosing a certain method over another one, if that's something you need to communicate to your readers. So let's look at organization and subheadings. So this comes from an article in PLS 01. So these are some sample subheadings under materials. So there's 4: study sites and experimental design, measurement and modeling of soil respiration rates, sampling of annual above ground productivity, species richness, and standing root biomass, statistical analysis or analyses. And that's actually a very common heading. I see in a lot of method section. There's always one on the statistical analysis used for that particular study, and that's always a good thing. It's always a good one to include. So 4 straightforward subheadings that really kind of walk the reader through just by looking at the headings how the study was actually organized, or how the authors came to the results through their methods. This is another example of some sample subheadings. A lot more. It's a total of, let me see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 11 subheadings. So a little lengthier, but it's okay as well. And this is also from PLS 01 on HISV infection, on effect on short-term NK cell memory. So the first section has ethics, mice, meaning what was used, HSV2 infection immunization, NK cell depletion, tumor challenge, mutation, blah, 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 all the way to the very end, data analysis, statistical analysis. Those are basically the same terms. But if you look at these very detailed subheadings, you can get an idea, even not knowing anything about the content or the subject matter. You can get an idea, okay, there are certain categories that were really important in the study, and they find themselves in these subheadings in a method section. So very detailed, very good overview of how the study was, how the authors designed their study and came to their results ultimately. So I mentioned earlier you want to cite commonly used methods. So here are some examples. This first one here, the detailed methodology, description of the service and the outcome measures, and the interview questions have been described in previous publications. Now this is passive voice, right? So it's perfectly fine. You can also do this in active voice, as this other article does. We use the same standardized protocol for 1H, MR, 2D, spin, so on and so on, as described in detail previously. Okay? So again, active voice, passive voice, either one of these options is fine. I'm always a big fan to use as much active voice as possible. In the method section, however, I tend to be more forgiving about a lot of passive voice. And so are a lot of editors and reviewers as well. So this is the flow diagram that I mentioned to you again as part of trying to save space in your method section. This is from the drug safety study that you guys looked at the intro for in the introduction video. So great detail. It describes, how many patients, over 1 million patients were originally recruited. They ended up with so and so many patients here, and it describes, how people, were excluded. And so on, and then how they were divided in different groups. So this will save the authors a lot of space to write about, because to write about in the method section, if they had to put all of this in words, it would be very, very long. So a flow diagram can really explain a very complex process in a very quick, in a very quick way without having to spend too much space in your method section to explain it. So the question always comes up, what tense to use? You want to use past tense to describe what you did. So at this point, your study is over, right? You're done. When your reader will read this. So examples to data were processed, notes were isolated. Again, both times passive voice, but definitely past tense. You want to use the present tense when you describe how data are presented in the paper. So data are presented in the following way. So depending on the kind of conventions you use to present the statistical data that you're giving to your readers. So that's present tense. Everything else is past tense. Now, passive active voice, I mentioned this earlier. The development of which has been markedly promoted. So you have some active voice here. Our group was previously interested in. We found that. So lots of active voice in here. Every sentence has active voice in it. That's really great. As a reader, I respond to that a little bit more positively, or it's easier for me to read through that than passive voice after passive voice. So my challenge to you is really think about how you can include more active voice in your method section, but don't feel like you have to completely take out passive voice in that section. You can use it, but again, challenge yourself to be more active. The same thing here, just another example. Reducing agent oxidation of proteins can influence its active voice. Its reduction may be advantageously used. Passive voice. We were interested. Active voice. It was found. Passive voice. Were studied. Passive voice. Was reached. So you see there is a good number of passive voice in here, but then subsequent increasing of phosphine concentration led to the signal height reduction. Active voice, which is probably caused. Passive voice again. The arising GSH signal corresponds to, again, active voice, are shown. Passive voice. So again, I'm not going to focus on the content here so much, but just we look at the tenses, or look at the tenses. And look at passive versus active voice continuously, changing back and forth between passive and active voice when possible. And that makes for really good reading. Okay, so the next video, if you want to take a break, will be about the results section.